There are three main stages to get from food molecules to ATP: glycolysis, the citric acid cycle, and the electron transport chain. The first stage is glycolysis. The glycolysis phase takes place in the cytoplasm, which can be likened to the gel of the cell, in which the organelles float. During glycolysis, the 6-carbon sugar molecule, glucose, is broken down into two pyruvate molecules, which are 3 carbon sugars. Pyruvate is the main product, but there are also two molecules of ATP and two very high energy NADH molecules. The second stage is the citric acid cycle. This cycle is also known as the Kreb cycle and the tricarboxylic acid cycle. As the molecules move into the mitochondria, the pyruvate formed during glycolysis will lose its carbon dioxide and become acetyl-CoA. During this oxidation process, lots of energy is released and then stored in two high energy products, namely NAD plus and FAD. The citric acid cycle takes place in the membrane of the mitochondria. Only small amounts of ATP is produced. However, it is the high energy products, NAD plus and FAD, that move into the next and final stage, where lots of ATP will finally be produced. The third and final event is the electron transport chain. This process can be further divided into three main events for better understanding. The first event is the oxidation reduction reactions. During glycolysis, synthesis of acetyl-CoA and Krebs cycle, the electron carriers NAD plus and FADH are reduced to form NADH and FADH2 respectively. These molecules are like little rechargeable batteries, and when NAD plus and FADH are reduced, this means that they accept and carry electrons and hydrogen ions, potential energy that can be used later in cellular respiration. In the electron transport chain, these electron carriers are oxidized, transferring their electrons to the carrier molecules embedded in the ETC membrane. In aerobic respiration, these electrons are passed from one carrier molecule to another in a series of oxidation reduction reactions, and ultimately to the final electron acceptor, oxygen, that combines with hydrogen, resulting in water, a metabolic waste product. The second event of the final stage is the creation of hydrogen ion gradient. The energy from each electron being passed down the chain is used to pump a proton through each carrier molecule from one side of the membrane to the other. This creates a proton gradient, a type of concentration gradient, and gradients are potential energy available for cellular work. The third event of the final stage is the phosphorylation of ADP. The hydrogen ions on the side of the membrane where most concentrated will eventually flow back across the membrane, down the proton gradient, through an enzyme called ATP synthase. As each hydrogen ion moves back across the membrane, the enzyme ATP synthase phosphorylates adenosine diphosphate to make the high energy molecule ATP, which can be used for many different energy requiring reactions throughout the cell. And voila! That's how living things create energy out of the food they get through aerobic cellular respiration.